Cuando se abra la presentación, ya se puede empezar. Bueno, buenas tardes a todos. El seminario de hoy va a ser más general, más de divulgación, aunque de todas maneras se va a tocar temas que tienen que ver con neurociencias. Quería decir que ayer se me olvidó mencionarles que allá abajo en el tablero de publicaciones tenemos un artículo de revisión que publicamos, mucha de la gente de la que ustedes vieron en las fotografías de ayer. Hay una revisión sobre la función, si alguien está interesado en verla, y bueno, los trabajos que él ha publicado, que yo he publicado sobre el tema, están a su disposición. Y el seminario de hoy, como decía, tengo que hacer otra vez la presentación porque como hay enlace por internet, entonces seguramente habrá más gente hoy que ayer. Quiero decir que el doctor Roger Traub originalmente estudió matemáticas. Después de estudiar matemáticas, estudió medicina. Después hizo su residencia en neurología. Entonces fue neurólogo un tiempo. Y desde los 70s su trabajo ha sido revolucionario porque él fue de los primeros que empezó a hacer modelos neuronales y modelos de sistemas neuronales y ha hecho varias predicciones gracias a sus modelos. Y actualmente trabaja en IBM en Nueva York, donde ha sido por muchos años, pues el que lleva, le preguntaba por qué trabajar en IBM si él no produce nada para IBM, porque IBM gana dinero. Y me dice que lo único que lo mantiene IBM es que pues, ha hecho algunas contribuciones que le sirve a IBM para presumirlo, entonces por eso sigue IBM. Pero tiene muchos contactos con muchos científicos, neurofisiólogos de todo el mundo, y ahora va a hablar de lo que usualmente habla gente muy importante después de hacer muchos experimentos y comprobar muchas cosas, es ya poder tener una visión más holística del sistema nervioso. Y la charla que va a dar es una charla para la que fue invitado como investigador Einstein, que es una fundación que da este tipo de nombramientos para poder ir a hacer estancias a Alemania. Y porque yo todo el grupo con el que él trabaja fue el mismo grupo de alemanes con el que yo trabajé hace muchos años y con quien sigo trabajando ahora. Entonces, bueno, Roger, thank you for your presence. Thank you for giving me this talk.
So, I understood 20% of that. The blue flag, okay. So let me give some background of this. A few years ago, I was this fellow in Berlin, and the foundation asked me to give a public lecture. And the only public lecture I've ever given before that was to kindergarten for my older son. And I took in a model of the brain and spoke to these kids who were 4 or 5 years old. I didn't throw anything at them. But this was different. This was to give a lecture to the Berlin public, and they wanted some kind of overview of what is neuroscience all about, and why do people do it, and how does the brain work, and all these things which are quite beyond me. So the compromise I made was, since I can't answer any of those questions, was why not? What is it that makes it so difficult? So some of this is in German, but I think I got it all in English. So if you think about it, there must be something like 50,000 or so neuroscientists on Earth, and they're all working, or many of them working very hard. And if you were to ask one of them, okay, so how is it that we can think, or how is it that we are who we are? I don't think you're likely to get a very sensible answer. So why should that be? I mean, it's not obvious that it should be like that. I just wanted to acknowledge one of my professors. This is Jerome or Jerry Lechman, who was a professor at MIT, where I was a graduate student for a year in the math department in the 1960s. And this man was a psychiatrist by background, and a philosopher, and a physiologist. He wrote a famous paper called What the Frog's Eye Tells the Frog's Brain, which anticipated Lechman, Maturin, McCulloch, and Pitts. And it anticipated Hubel and Wiesel in important ways. So I won't tell any more stories about him now. So this is a summary of what I was trying to convey in Germany. So the first point is, why do people do neuroscience other than to make a living? So the first thing is, we want to treat neuropsychiatric disorders, and schizophrenia, and Alzheimer's disease, and so on. We want to learn about the brain so as to imitate its functions. This is like the AI community is very involved in that, or at least they pretend to be involved in that. And then there's this philosophical side, which is to understand ourselves and our origins. I don't know how many people in neuroscience actually worry about this, but I know some of them do. So now, two is my view of what does this come down to? What does it mean to do any of these things? So, because of my background in mathematics and so on, I thought, well, that means we should be able to relate physical properties of neurons, something concrete, and the connections between neurons, and relate that to functionally meaningful circuit outputs. So as to say, we have the organism or some part of the organism, and it's doing something which we understand what it's doing as a behavior, and then we have this circuit that is generating the behavior, and we can account for in concrete physical terms. That to me is sort of the basic goal of what neuroscience should be. Now, the problem is that all sounds great, but how is it that you can actually do that? So what are and three is, what are some examples of where people have actually been able to pull this off? So one set of examples comes from analyzing small circuits, including so-called central pattern generators. And I'll show you some details of a central pattern generator from the lobster in a few minutes. So another case where you can actually account for circuit behavior in a pretty detailed way comes when you look at network oscillations and seizures. And the reason that that's possible is during these oscillations and seizures, what you've done is you've taken a physical system which has many, many degrees of freedom, in the sense lots of cells doing lots of things, and by putting a drug in the bath or observing a seizure, you've reduced the number of degrees of freedom in the sense that lots and lots of cells are doing the same thing at the same time, which means that you can make fewer measurements and then you can still make some physical sense out of what the circuit is doing. That's all great, except it's not functionally meaningful. So, then to extrapolate for cognition, this program, which I've outlined here, is proven difficult for conceptual reasons. We don't really know what we're doing. We don't have enough data, so on. But mostly, I think, it's the lack of a suitable preparation, biological preparation, where you can analyze the neurons, the circuits, and the relevant outputs at the same time. So, yeah, okay, this is something that should be obvious, but somehow it's not. Like I remember sitting at IBM a few years ago, and DARPA, some idiot from DARPA, that's the Defense Advanced Research Projects Agency, is connected with the Defense Department in the US, and they like to support way up projects, and they were going to develop some technology that would allow them to measure simultaneously 1 million neurons. If they were in 1 million neurons, and they had the idea, if we can do that, then we understand everything. And that is so utterly stupid that one doesn't know what to say. So you think this is obvious, but it's not obvious. So now, a little bit of self-indulgence. That's my grandson, Isaac. So Isaac sort of set it all. So this is when he was 3. For some reason or other, I don't know why, I asked him, why do we have a brain? And he said, to my surprise, so we can think, which is a pretty good answer. And then perversely, I asked him, that's fine, but what was it about the brain that makes it possible for us to think? Which of course I had no idea myself. And he didn't have an idea either, unfortunately, and he changed the subject. I should just stop here. So what is it that makes it so hard for us? One is how many cells there are. This is, Farid was a former student. He's now a neurosurgeon. So he made this lovely Golgi impression of stain of cortex. He was going to publish an atlas at this point. I don't know if he ever did. So this is staining, I don't know, 1% or so of the neurons, and it just gives some, looks like a work of art, but some sense of anatomical complexity. This is a beautiful picture from Ramon y Cajal. It's got this stamp from this museum in Madrid. They very kindly let me reproduce this. But it also is just to give a sense of the complexity. Probably, I don't need to do this, everyone knows this already here. But just for example, anatomical complexity of single cells, like this Purkinje cell, but also the variety of cell types. Comparing this thing with the granule cells, for example, of which there are oodles and oodles. So that's part of the problem. Let me digress for a second. Does it mean we know nothing? And just a little bit of history. people have been thinking about these sorts of things for a long time, long before people knew there was such a thing as a cell, or any, let alone properties of cells. This is an example I got from this nice book. It's an illustrated history of brain function, and it sort of anticipates fMRI by a few hundred years. This is from about 1497. But what it tells you is people then, and way before that, were thinking already. First of all, thoughts come from here, and not from the gluteus maximus or from the belly button, but from here. That's already a big step. And also that maybe different parts of the brain did different things. Now, how it is that people have that idea, I don't know. That the idea that thinking is connected with the brain at all, I think was a big advance. And maybe it came about from studying effects of head injuries, or noticing that if somebody was clunked on the head, they would become unconscious. Maybe that was what led them to that notion. I mean, we take it for granted. You should ask yourself, where does this assumed knowledge actually come from? So another source of knowledge about brain function came from the clinic. So in the 19th century, people began to correlate pathology in tissues with manifestations of disease. And in the case of neurology, Paul Broca and others started to correlate stroke lesions and brain tumors in particular parts of the brain with particular deficits. This again is an enormous conceptual advance. Just noticing that, this part of the brain would do this part of the brain would do that. The left hemisphere controlled the right side of the body, and so on. And this became more and more detailed, and then people started with the developments in staining tissue and in microscopy. People they noticed that there were cytoarchitectonic differences, but also functional, sort of very detailed functional correlations between what would happen along the motor strip and some manifestation, say seizures or paralysis in some part of the body. But this is from the early 20th century. Notice there's a lot of detail here, but there's nothing here. It's sort of empty spaces, most of the temporal lobes and frontal lobes. So another strand came from psychiatry. This is psychiatry without neurons. Sort of how it is phenomenological psychiatry. So I took this exchange from a book on schizophrenia by McKenna. I'll just read it. So this is the psychiatrist. Have you been nervous or tense lately? The patient, no, I got a head of lettuce. You got a head of lettuce? I don't understand. Tell me about lettuce. Well, lettuce is a transformation of a dead cougar that suffered a relapse on the lion's toe. So even my beloved president, however incoherent he is, doesn't sound like that. So
It sounds worse.
it's worse in some ways, because I think this guy wasn't able to destroy the planet, but was probably confined somewhere. Anyway. I don't know what to say in terms of explanation. It's more defining a problem. So maybe I've left a sense of how difficult everything is. But now here's an area where things are not quite so difficult. So this is what to me is one of the great success stories of neuroscience, and it has to do with with crabs and lobsters. And much of it is connected with Eve Mortar, who I guess, is it you?
No,
You know who?
we don't talk about chicken from.
Okay. So just a background. So our teeth are in the mouth, which is pretty well known fact. But in a lobster, the teeth are actually inside the stomach. So the lobster kind of feeds on the bottom of the sea, and they take in bits of this and that, and go into the stomach. And the stomach has 3 teeth. So I guess this is one, and that's the third one. And the food is actually ground up in the stomach. And it's been possible to actually put an endoscope into the lobster's stomach and observe the different motions that it produces. So at that level, there are just a few different ways that the stomach actually grinds up food. The question is, how is it controlled? How are these motions controlled? And the analogy I have is like with a washing machine, which does a few different things. It fills with water, it can spin, and it can rotate, and it can empty the water. And this is something like that. And you have a little computer which controls what type of motion is being produced inside the machine. So the lobster has something like that. And yeah, so this is kind of the front end of the lobster, and that's the mouth. And the stomach is in here someplace. So the different motions of the teeth are controlled by a relatively small circuit, which looks like this. So this has the advantage that you can take the animal and actually remove the entire circuit. So you sort of cut these nerves here, and you have this group of cells. And you can actually see the cell bodies there. And the lobster also cooperates with the scientists in that many or most of these cells are identifiable, so that they have very similar properties, location, size, whatever, from animal to animal, which makes it easier to collect the database, since the circuit is relatively stereotyped from individual to individual. So the anatomy of the individual cells is actually horribly complicated. So these are fills of some of these cells. And you can see this enormous mass of neurites, but from which it's impossible to record. But the cell bodies are very large, and it's quite easy to record from them. So if we just pretend that we don't care about what's happening out here, which is an approximation, then what can you do? Well, because there aren't that many cells, and because they're large, and because you can see them and identify them, it means you can record from each one, and you can record from all of the pairs of cells. And that means that you can characterize the biophysical properties of each neuron, and you can characterize the connections between all pairs of cells. So this is a diagram, sort of a schematic diagram of that. It doesn't tell you the intrinsic properties of the cells, but it sort of sketches the connections. So like this resistor is a gap junction. So there's a gap junction between these two cells, and then there are excitatory and inhibitory connections between other pairs. Some of these connections are graded, so there's transmitter release that's not action potential control. And I forget what how this is shown. But some of them are action potentials and some of them are not. So this is what gives an idea of what the circuit actually does, is it makes ongoing oscillations. So these are membrane potentials of different cells, so the individual cells oscillate, and this one's not firing, and this one is. And the important thing is that there's a period for these oscillations, there's the relative phase, there's the intensity, and that then, because the connections are known between this circuit and the muscles, it means you can relate the behavior of all of the cells to what the stomach is actually doing. And then in addition, you can put various so-called modulators in the back around this. This can be done all in vitro, and you can convert one sort of pattern of oscillation into another. And you can then analyze how that works. Is the modulator acting on the connections, or is it acting on the membranes, and so on. Okay, so this to me is like a model of what we would like to do with ourselves, because you have the anatomy, you have the physiology, you understand the biophysics, and you can relate the behavior of the circuit to the behavior of the animal. Somebody's interrupted. Okay. If only life were so simple. So, if you try to do this at a higher level, of course, it's not so simple, because again, so many more cells, different types of cells, and the difficulty of relating the physiology of the circuit to the behavior of the animal. So I just picked a couple of examples which have the flavor of the stomach gastric ganglion, but are also different. So this one I like, but it has not been investigated very much. So there's a group of fish, a weakly electric fish. Some of them live in the Amazon River, sort of at the bottom. It's kind of muddy and murky, and the animals, I guess, have trouble seeing each other. But they communicate by producing electric fields. So each animal can produce an oscillating electric field that it senses itself and that other animals can sense. And these fields oscillate at very high frequencies, like hundreds of hertz or even over a kilohertz. So there's a central pattern generator for that, which is found in the medulla of the fish. It's called the medullary command nucleus. It has roughly 200 cells, 40 big cells like this, and a bunch of smaller cells. And this is of interest to me because this circuit is full of gap junctions. But there are only a couple of papers about the physiology of this. I don't know why that is. Maybe the animals are hard to get. So another relatively reduced system is the in vitro spinal cord. So this is an example of that. This horrible thing is a lamprey. It's a jawless fish. So this fish has a jaw. This one doesn't. It has a sort of sucker, and it's a parasite, and it's sort of attached itself to this fish here. So these animals, the tissue has a property that it survives anoxia very well, and you can remove the spinal cord and have a preparation consisting just of the spinal cord and maintain it in vitro and do intracellular recording. And the behavioral correlate that people look at is, for example, Sten Rilner in Stockholm, is swimming. So you can observe the animal swimming in its natural state. They can swim forward, they can swim backwards, they swim at different velocities. And in the isolated spinal cord preparation, the animal, of course, isn't swimming, but you can record from all of the motor routes, and you can predict from the signals in the motor routes what the animal actually would do. So it's called fictive swimming. It's not moving, but it's generating neural outputs that would produce swimming of one sort or another. So this is showing how neural activity kind of goes from one end of the spinal cord to the other, and this is what produces this sort of wave of activity that generates swim. And people have looked at what controls the frequency of this oscillation. Does it go forwards or backwards, and so forth. And there are some very detailed models of this behavior. Now, it's not, of course, not the level of somatic gastric ganglion, but surprisingly detailed. Okay, now can we go to the cortex? Well, so yeah, I've shown number of cells, give a sense of number of cells, different types of cells. Here's another type of complexity, and that is neurons, contrary to what some of my colleagues back home believe, are not points, but they're distributed in space, at least a lot of them, some of them over quite long distances. This is 100 microns, so it's more than a millimeter. So it's a layer 5 pyramidal cell. And the problem here is, it's not just that it's big and has lots of inputs, it's that the properties of the membrane in different parts of the neuron are very different. This was been known for a long time. One of the pioneering studies of this was from Rodolfo Unas in the 1970s, maybe even earlier. He recorded from alligator Purkinje cells and was able to record from both the cell body and from the dendrites. And he showed that the cell body would produce fast spikes, sodium spikes, and the dendrites would produce slow calcium spikes. And this is not taken for granted, but this principle has been discovered and rediscovered and rediscovered, and this with better and better techniques. And this shows it with patch recordings from Bert Sackmann's lab long after Rodolfo's studies. But they were able to actually visualize the cell and the preparation simultaneously, patch on the soma and from the distal apical dendrites. And sort of this pyramidal cell is doing something at least qualitatively similar to the Purkinje cell, so that the soma is generating fast spikes, and this part of the apical dendrite is generating this slow calcium-mediated spike, which you see here also. So this is 10 milliseconds, so this is very, very broad. And then it doesn't show the axon. The axon has its own distinctive properties. So putting this together and trying to understand what this single cell does is not so easy. Even not, let alone not doing it quantitatively, even doing it sort of quantitatively is very, very difficult. Not one cell. So is it all hopeless? I hope not. So this is an approach that kind of I had based my career on, which is, okay, we can't study these very large normal circuits doing their normal thing and relating them to behavior. But as I said earlier, what you might be able to do is take a preparation, poison it, not kill it, but poison it, and just simplify it. So this is not a totally crazy thing to do. People have sort of observed in experimental models of epilepsy that during this so-called interictal spike, EEG spike, if you record an EEG spike that's, say, generated here, and you record from the cells in that focus, there's an enormous amount of redundancy in the behavior of the cells locally. And with the development of the slice preparation, and then using first in the liver and then in brain, and then Per Anderson and David Prince started showing that you could generate epileptic-like behaviors in the slice. Now in the slice, in one region of the slice, you only have thousands of cells as opposed to tens of millions. And because you poisoned the slice and produced a strange condition, there's extreme redundancy in what the cells are doing. It's not total redundancy, but it's very large. So this was an example of that kind of thing. This is experiments that were done by Bob Wong starting in the late 1970s when he was a postdoc with David Prince. So what you see here is he's got the slice sitting, It's GABA A receptors have been blocked, and here is recording the field potential. And this is a local stimulus that he used with either a current pulse into the tissue, or he would puff on a little bit of potassium chloride. And what he got was something that looks like a physiological shock wave. For people who were here yesterday, there's a slow wave that lasts about 50 or 100 milliseconds, and then there's a superimposed ripple. This is well before the ripples were discovered in shock waves. That was 10 years later. But this was known as to occur during epileptiform events, usually for a long time. So there's synchronization of activity at two time scales. So this time scale and this time scale. So now if you look at what the cells are doing during that, most of the cells are doing this. They're making bursts of action potentials, and this is a calcium-dependent event here, and this. But this is also synaptically driven, and the way you can show that was you produce the same event, but you pass a big current through the cell, so it's very hyperpolarized. And then during this event, you see a huge EPSP, or excitatory synaptic potential, which tells you that this cell is receiving lots and lots of inputs, and that's also contributing to this envelope. So that was well known. What wasn't understood was that how is it that this synchronization comes about? And that turned out to be historically important because it led people like Ed Dudek to ask, first of all, how are these cells connected together? So in the early 80s, there were the initial studies of the actual connections between pairs of these pyramidal cells using two electrodes. Ed considered the possibility that gap junctions were important for this synchronization. And it turns out that they are, but not in the way that Ed thought. And this is what I talked about yesterday. So they're important for this high frequency synchronization, but not necessarily for this synchronization. So what next? So that came from Bob's experimental intuition. He noticed two things. One was that after a stimulus, there was a latent period before the population event occurred. And this latent period here is about 50 milliseconds, but it could be as long as 200 milliseconds. And the question was, he's given a very brief pulse. What is it that's actually happening there? And then he noticed, just by recording cell after cell after cell, he noticed accidentally that some of the cells would produce bursts during the latent period. So, so that was a clue to kind of the, I would say, the basic physics of what must be happening. So I had developed a model of bursting in cells, which is it's all wrong, but never mind. It looks like a burst or like that. And the question is, what kind of simple model would account for these observations? And it turns out it's completely trivial. If you just have lots of cells and you connect them with synapses with a property that each neuron connects to more than one other, and that a burst can propagate from one neuron to a connected neuron if inhibition is blocked, that is sufficient to account for these observations. And this is sort of a model demonstration of the similarity that you get. So this is a network of cells connected with this principle, with intrinsic bursting and so on. You stimulate a few cells that do this, kind of like that, and this propagates from cell to cell, and you get exponential growth of behavior. The whole system goes off, and because the cells are firing simultaneously, you get this giant EPSP, and it all worked. And it made the specific predictions. I don't know. There's a slide. No, I don't have the slides here. The specific predictions were that bursting would propagate from one cell to another. That was actually shown by Richard Miles a couple of years after this. This also predicted you should be able to stimulate one neuron and set off an epileptic event. And Richard Miles and Bob Wong showed that also. So what this model doesn't account for is the ripple. But that's another story. So anyway, this is an example that if you have, even though it's a mammalian preparation, because it's reduced, it's possible to make sense in sort of some physical way of what the cells are doing. What you can't do with this is explain anything about behavior. You can provide constraints, like people make these models of associative networks in the hippocampus, but I don't know how often they take into account the actual properties of the synapses. So here's another example. Purely by coincidence, these examples are all coming from my own work. So here, this is Miles Whittington. He's looking at oscillations in a neocortical slice preparation in layer 5. And he made, I think I showed this yesterday, but not in such detail. He made the tissue alkaline, which presumably is opening up gap junctions. And what you see in the basal conditions is oscillatory behavior. This isn't showing the field, but this is oscillatory. It's a mixture of these spikelets and action potentials. And when you alkalinize the tissue, it becomes much more intense. And this is consistent with gap junction coupling. And again, in this kind of behavior, you can account for a lot of the observations with models. Now the models, in one sense, are complicated because there are all these membrane channels and kinetics and blah, blah, blah. But the actual principles that are involved are extremely simple. And it's namely spikes propagating from one cell to another. Extremely, extremely simple. So what does that matter? Well, this is kind of reducing the degrees of freedom, but in a patient. So this is it's called electrocorticography, surface electrodes. This is from a child who had intractable seizures. She had a cortical dysplasia in a frontal lobe, and she was being evaluated for surgery. So this was in London, and they did this recording, and they captured the onset of one of her seizures, which would be this. So that's called an electrographic seizure, and this would have a behavioral correlate, at least in her. And what Torsten Baldeweg and noticed was every one of her seizures began with this high frequency oscillation. I think I may have showed something like this yesterday. So from what Bob and I were doing before, we can account for at least some of the properties of these waves, and then kind of what I was talking about yesterday, we can account for that, at least up to a point. And then this is, I think, these two slides will be the last example of this kind of thing. This is, well, again, it's a slice, so you vastly reduce the number of cells as compared with the whole brain. And in addition, there's a drug in the bath. So it's producing an artificial condition. It has the advantage that you can study it experimentally, and it has a disadvantage that, how do you relate this to behavior? But to some extent you cannot. If I remember, I'll try to point that out. So this is actually, it was a remarkable experimental observation which came quite by accident. So this is kind of a diagram of the slice. So this would be the superficial layers of cortex, and this is deep layers. And then this is either layer 6 or subcortical white matter, I forget. So Miles put this drug in the bath, it's called kainate, and he observed that it was known that kainate would induce oscillations in brain tissue, but what was remarkable here was that you get different oscillations in different cortical layers. which debunks the whole notion that cortical columns are doing somehow uniform things and that they're defined entities. So in this case, this part of the column and this part of the column are doing different things. So specifically, this is a time frequency plot. So this is 20 seconds. And this is recording superficial layers. And the red is kind of high energy. So what you see here is a sort of 40 hertz oscillation, gamma frequency oscillation going on superficially. And whereas in the deep layers, you see a 20 hertz oscillation, or 25 hertz oscillation, beta 2 frequency. These are kind of what they look like, what the field potentials look like. So Miles then was able to make a cut. I don't know how he did this with a little razor blade. He made a cut to layer 4. And he showed that the superficial layers on their own can do this, and the deep layers on their own can do that. So this is very, very striking. So then, but the nice thing is, it's an oscillation, there's stereotypical behavior, it's a reduced preparation, maybe we can figure out what's going on. And it turns out what's going on was very, very odd. What it is this oscillation doesn't need chemical synapses. And in addition, if you record from an individual cell, what you see, mixture of spikes and spikelets, an anti-German spike, so you can see the notch here, and the spikelets tended to occur in pairs. So again, by just taking model pyramidal cells and connecting them together, not having synapses but having electrical coupling, you get this behavior. It looks incredibly like the experiment, and it made it again specific predictions, which you worry about. What is it that determines the period? That's always with an oscillation. That's what you want to know. What determines the period? And it's not a synaptic current, so we guess it's an intrinsic current. And as it happens in the model, this period was determined by the kinetics of a potassium current called the M current, and that turns out to be true experimentally also. I think that's an accident, but it turns out this period is determined at least largely by an intrinsic current. So this is from an association cortex, but it turns out that in behaving humans, you actually can observe oscillations at this frequency that correlate with motor behavior. What you can't do is show that they depend on gap junctions and not on synapses. At least I don't know how to do that. So there's potentially a behavioral correlate, but we don't know how to carry it out. So the last experimental slide I want to show is sort of another way of looking at the nervous system, which is a more kind of cognitive / single unit recording, or nowadays multi-unit recording. So this was from the work of Patricia Goldman-Rakich, who actually died in an auto accident some years ago. But this was a classic study. And so to understand this, we picture a monkey, a rhesus monkey, who is awake and is actually part of the experiment, so it's cooperating with you. And the monkey is sitting in a chair, and with its head is fixed, and there's a computer screen in front of it. So, and then the experimenter controls what is on the screen, and the monkey can either do something with its paw, or it can move his eyes. So there were two kinds of paradigms. With one, the experimenter would the monkey would fix here, and then the experimenter would have a light shining someplace, a flash of light, in this case there, or there. And if you record from the right part of the frontal lobe of the monkey, you find neural activity that correlates with the direction. So the task the monkey had is fixing, light goes off, the light goes on, light goes off, a few seconds, light comes on again, and the monkey has to say, oh, it's the same direction as the first one, or it's a different direction. So that means this is a test of working memory. It means that the monkey has to retain some notion of where the first light was so that he can do, or she can do, a comparison with the second light. And so what you see is something very interesting. Here is before the light comes on, goes off, then there's all these spikes, there's all this neural activity for, in this case, 3 seconds, and then the second light comes on and the animal does something. So what it means is that there's a physical correlate in the brain of working memory, which you can, in principle, which you can observe experimentally. It's pretty amazing. And I asked someone how long this can go on. The record, he said the record was 10 seconds or so. So this is like what you do when you're given a phone number and you have to remember it long enough to actually dial the number. That shows my age, dial the number. So anyway, working memory. And it turns out, you don't see it here, but it was later shown that this is oscillatory activity. So you see gamma and beta oscillations during this working memory. So in this task, it's analogous, but here the recording is from inferior temporal cortex and lateral temporal cortex, and I forget which, or both. And instead of having these lights, the animal is shown pictures. So here's a picture of a monkey and some more abstract stuff. So there's a flash of the picture, it goes off, the monkey has to remember, flash again, is it the same or is it different? And again, you see this continuing activity, sort of this neural correlate of working memory. Of course, no one really knows what the basis of this is, what the physiological basis is. There are some theories and models, but as far as I'm aware, it's not really known how this is coming about. Okay, this just gives a sense of the direction of how you might be able to relate sort of studies of oscillations like that with something behaviorally relevant like this, but it's very hard to prove in vivo that the mechanisms, the cipher mechanisms here, are the same as what you see in vitro. So, is there a mathematical shortcut to all this? If all our neurons were the same, had identical properties, and if they were all really simple, either of which is true, we could borrow ideas from statistical physics, as people have tried to do. I probably shouldn't talk about this. I'll say something that I will regret. But this is, I don't think, I mean, this is kind of nice at the moment, but in the long term, if we think of the long term of where neuroscience is going, I don't think this kind of thing is going to help too much. But maybe I'll be wrong. But, okay. So this is just kind of summarizing. We want to better treat neuropsychiatric disorders. A lot of what goes into that now is phenomenology, which is okay. It maybe is the best we can do. Really being more ambitious than I ought to. Learning from the brain so as to imitate its functions. This is something where I have to really watch what I say, because I'm part of artificial intelligence at IBM. So I have to maybe change the subject about that. And understanding ourselves and our origins is something I think is also, at the moment, somewhat intractable. I gave you an example here, the somatogastric ganglion. And here is somatogastric ganglion, which shows some oscillatory behaviors and seizures. This is sort of a feel for these, how I see things. And this is obvious that this next step is hard. It's going to be very hard. This I showed yesterday. I like this picture. There's a question, what makes us human? Something that this guy thought about. He's a very strange looking guy. I guess he's human. But the puzzle, the reason for showing the neuron here is that if you look at our neurons like this is a human neuron. If you look at a rodent neuron, I think this is from a rodent. They don't look that different. So I better stop. So this German sort of now where, and this is a quotation from I guess he was an Austrian philosopher, Ludwig Wittgenstein. And what this means is what you can't talk about, for those things you should be quiet. So thanks.
¿Alguien tiene alguna pregunta o comentario? Y sí empiezo yo. Bien. Would you think that there would be a maximum number of cells that we can record so that we can make some predictions from actual cell recordings? Or it's a more modular thing so that you can find instead of 1,000 cells and record it, then at the same time you would find.
Yeah, so this is how not everybody agrees with this, but the thing here is it's not just recording from the cells, it's knowing the structure of the circuit. And that's what was missing with this DARPA engineer was you can record from as many cells as you like, but if you don't know their properties or how they're connected, it's impossible to make any sense of it. So the thing is, I don't know what preparation has sort of the right number of cells, so it's complicated enough to be interesting, but still simple enough where you can sort of figure out its anatomy. And that's kind of the conflict. But that's how I see it.
And how much of a problem these difference of concepts, for example, functional connections versus anatomical connections brings? Because you know when you analyze functional connections, you try to relate the activity of one cell with another cell, but you don't know what happens in between.
Yes. Right.
That versus true direct contact.
Yeah, so again, this is observing correlations in cellular activity in vivo, like sort of thing Bujaki does. That's an important step, but so much is missing in understanding, how is this happening? It's the same thing with the Goldman-Rakesh stuff. It's very interesting phenomenon, but we don't know how it's happening. And if we don't understand the mechanisms that underlie it, we sort of are stuck. We don't know where do we go from there. How do we manipulate it? How do we make more predictions and so on?
Yeah, it should be by now quite apparent that thought is an emergent property of these networks. So this is a question that has to do about your experience feeling about this. So at what level does this emergent property comes? At level of some dozens of neurons or hundreds of neurons?
You mean sort of interesting emergent, not epileptic, but sort of interesting emergent properties.
Yeah, exactly. It's a collective property of these networks. And at what level of complexity should we focus our efforts? Because I think that
I think that's sort of a question for natural history. Yeah. I mean, if you look at bumblebees or termites or insects, there's a lot of very rich behavior that they do.
Yeah.
I mean, it's not completely rich because sort of when you get into it, a lot of it's actually quite stereotyped. But even so, it's very, very rich. So there you're talking about, I don't know, 100,000 cells. I guess in a honeybee is a million, something like that. Cells.
Minerals, they have fewer. Yeah?
So,
They have cells in total.
total, yeah, that's total.
Organs.
Yeah, so, I mean, I used to make jokes when people made slices, so you have a few thousand cells.
Yeah.
So then, and then here we have, I don't know, 10 billion or 100 billion or something. So think of intermediate scales. So Rodolfo Linas had this isolated brain preparation. So it's like the brain removed, functionally removed from the animal, perfused with some solution. And this raised a lot of ethical questions. Like, is it proper to make a preparation like that in a mammal? Okay, so I don't know the answer, but if to say it's proper, it's not proper, but is it proper to make a slice? There are thousands of cells there.
Yeah.
There is emergent behavior. It tends to be simplified. Or maybe we're not looking at it the right way. So I've thought about this, but I don't know the answer. It's like there's some continuum, and I don't know where you draw the line. But it's an interesting question.
I have a question. Somebody will probably naive with phrases.
We're all naive.
So, for example, the rodent hippocampus is huge compared to the human hippocampus, right? But
You mean relative to the size of the whole brain?
it's huge, and the synaptic complexity of the rat hippocampus is huge.
Yeah.
The memory processing of rodents is, I think, not compared to human memory, but again.
Yeah, so, maybe the hippocampus is a preprocessor. Something computer science jargon, but it's a preprocessor, and then it's kind of all the rest of the brain is really, is really good. That sounds very superficial, but maybe there's some truth to it.
All right.
How do you think the networks function working in brain plasticity?
Yeah, so let me go back to Terry. So this is one quote I remember from Terry. He said, "There's no chance of understanding learning in a system if you don't understand how it works without learning." So I'm punting. I don't know how would you say that? It's a sort of slangy term. I'm avoiding your question. I don't know how to think about that since I don't understand the kind of basic operation to begin with. And this is an idea that I think is completely lost in the artificial intelligence field. There's so much focus in deep networks on how they learn, but there's very little understanding of how they actually work. The complicated ones. So I'm sorry.
I'm Elsie Rocco. I'm across the way in educational research, so I'm very naive. But I was interested in your talk because in educational fields, neuroscience has become so important. Teachers are being trained in some version of it, as though it's going to be the solution for the future of educating children.
I don't believe it for a moment.
Good, but I need arguments. Because I don't, I really respect the science of it, And, who's making that jump in that resource? Is that part of
I could tell you a story.
Sure.
So when I was an undergraduate, the valedictorian of the class was brilliant math student, absolutely incredible. And he gave his valedictory speech, and I'll summarize it. It was that we understand a lot about optimization problems and from pure mathematics, and we can apply that knowledge to international politics. And by looking at things from game theory and from convex optimization, I don't know what, I don't remember the details of this talk, but the extrapolation was he's going to solve all the problems in politics and international relations. And there's a dead silence in the audience. And I think you sort of get the point. I think it's like that. How are we going to make this jump?
But it's happening.
Whereas education, as I understand it, is very pragmatic and practical, and you kind of see what works and what doesn't work, and does it need a theory?
Well, we do research. And we do
No,
have
I mean,
theories,
yes, doing research is fine, but having
I don't know exactly what the basis that they're using
Yeah.
to train teachers in this. It seems very far off. And it's pushing other kinds of research off the map.
yeah, it doesn't make any sense to me. The same kind of thing is happening in psychiatry. A lot of very ambitious claims are made based on, I don't know, properties of semantic plasticity or this or that in the nervous system. This doesn't make sense to me. That's not an argument, but that's all I can say. Have we got any more controversial?
Well, just a commentary about it. I think this jump is like jumping across many levels of emergent properties. It has to do with many levels of higher and higher levels of complexity. So it's like to claim that from basic physics you can understand human behavior. It doesn't make sense. It doesn't exist at that level.
That's right. That's exactly right.
Okay, thank you very much.